Material information. Can there be a physical entity that is the content and also the medium of its information as a whole? Let me try to elaborate on the question. Assume there is an object X, and the object's information constitutes the object's physiochemical properties. That information should be communicated to any surrounding entities that can be perceived. Assume there is a mold to make an idol. The mold should be an engraving of the idol it needs to make. There is information transfer. That is, the mold dictates the shape of the idol, but the information of the mold is kind of minus, negative, to which the idol is a plus. The information of the idol made as a shape is opposite to that of the mold. If a mold can make another exact copy of the mold, then that is absolutely information transfer. There seems to be no way for one entity to create another entity with the same information. There is no scope for self-replicability. In another way, take a pair comprising a mold and an idol complementary to the mold. That pair together can be a unit of reproducible information because they can make more pairs together. The mold can make more copies of idols, and the idol can make more copies of molds. That way, one pair of positive and negative can make many more identical copies of the mold-idol pairs. Replication of the identity of an entity is almost impossible unless there is some way for the information to be transferred from one entity to another. That is what life is. An organism makes a copy of itself. That is the unique property of living things. Living things make more copies of themselves by themselves, autonomous self-replicability. What makes this possible? It is made possible by the genetic information in all living beings. It is information technology in biology, the bioinformation. To me, our ability to understand bioinformation is the most outstanding achievement. Information is a sequence of subunits with size and ordered variables that can be distinguished from other sequences. That physical entity should have discernible information, and it should be replicable. The nature of bioinformation. All the information in biology is a sequence of nitrogenous bases in nucleic acids, DNA and RNA, and a sequence of amino acid residues in proteins, which also depend on the former. The base sequence dictates the amino acid sequence, and the amino acid sequence dictates the structure of proteins. The structure of the proteins dictates their interacting partners and thus functions as enzymes or structural proteins. Eventually, the proteins dictate the formation, maintenance, and propagation of cells' organisms. The genome is incredible in that all the events, functions, and properties of the cells and organisms are encoded in the form of the base sequence or a nuclear program. We know a lot about the nature of bioinformation, how it emerged, propagated, and evolved. The information system in biology has a material form. The transition point from matter to matter with information is an open question. Unlike abstract languages that can take the form of sound, biological information is physical. Bioinformation is composed of matter and codes for matter in a specific structural form. All parts and the entirety of an organism are manifestations of the bioinformation within. A physical entity must possess 3 critical properties if it is considered a molecule of information. These properties are I, diversity in the constituents of the molecule, T, a specific sequence of the constituents, and, most importantly, D3, the ability to replicate the information. If we require a molecule to have multiple repeating subunits, it must be a polymer. Carbohydrates, proteins, and nucleic acids can form polymers in biology. Lipids can form structures or conglomerations, but are not covalently linked subunits to each other, and therefore cannot be called polymers, at least for the sake of our discussion. Carbohydrate polymers of glucose include starch, glycogen, and cellulose. However, their monomers do not exhibit diversity to create complex information. Proteins are polymers composed of 20 types of amino acids. Proteins are made up of diverse monomers and can form very long chains. Therefore, proteins are molecules with information. However, the essential criterion is the replicability of the information. A protein cannot dictate the formation of other proteins with the same sequence. Nucleic acids are polymers made from 4 nucleotides, each consisting of 1 nitrogenous base, adenine, guanine, thymine, or cytosine. These nucleotides are analogous to the alphabets in a language. There are 26 English alphabets with which we can create an almost infinite number of words with varying numbers of letters and combinations. Theoretically, if each word consisted of only one letter, we could make 26 words. If each word consisted of two letters, we could make 676 words, 26 squared. If each word consisted of three letters, there would be 17,576 possible combinations and 456,976 possibilities for a four-letter word. For perspective, there are 6.15 by 10 quai 6 possibilities if we could create words consisting of 26 letters each. Not all combinations of letters are useful, but these examples help us understand the significance or relationship between the number of unique letters, the number of letters per word, and the diversity of words that can be formed. The information in DNA, for example, should eventually code for proteins made of amino acids. DNA is made of only 4 different nucleotides, while proteins are made of 20 different amino acids. If the logic for nucleic acids is to count the bases in twos, that is, as dinucleotides, in that way, the diversity can be increased from 4 bases to 16 different dinucleotides. That is insufficient in a way. Then, one can increase the subunit size by something like a trinucleotide. If the DNA sequence was read in steps of 3 bases, one could generate 64 trinucleotide subunits. That is what all the protein coding gene sequences are read as. They are read as continuous steps of 3, and each is called a codon. One codon codes for an amino acid. Very similar logic also happens in the binary computer language. Each bit can be either a 0 or 1, but if we put together 8 bits, we get a byte. If each bit could be either 0 or 1, and we put together 8 of these, then we could have 256 diverse bytes that can be used to write a script or a program. Let me try to use the English language as a medium of communication with bioinformation, a comparison of languages. The English language has 26 alphabets, A to Z. In comparison, the bioinformation language has only 4 alphabets, A, T, G, and C, representing the nitrogenous bases in the nucleotides that make up the nucleic acids. These bases should be held in position to represent a specific sequence, which would otherwise be a constantly changing sequence. As nucleic acids, these nitrogenous bases are held in sequence by a phosphate backbone consisting of alternating phosphate and deoxyribose, or ribose in RNA and DNA. The word size in English is not fixed. Some are very small, while others are lengthy. The longest English word is pneumon-ultramicroscopic silicovolcanoconiosis, the name of a lung disease caused by the inhalation of excess silica dust from volcanoes or other sources. Most of the longest words are names of several diseases. One interesting word is supercalifragilisticexpialidocious, which is a phobia of long words. In the language of bioinformation, the word size is fixed to 3 alphabets, called a codon, none smaller or larger. Another critical difference is that there are no spaces between the words in bioinformation. All the codons are read continuously. The start and stop of a sentence in English are distinguishable. The starting word has a first capital letter, while the end of the sentence is denoted by a period. In bioinformation, the start is typically the ATG, the start codon, AUG in RNA, and the end could be TGA, TAA, or tag codons, the stop codons. The sentence length in English and bioinformation is unlimited. Therefore, very complex information can be generated from either of the languages. But the kind of information is different. English generates information that is abstract, immaterial, and non-physical. The meaning of the English sentence can only be perceived by the minds of the people who can understand English. The information in biology is physical. Each of the codons codes for an amino acid. 20 amino acids are naturally occurring in proteins. There are 64 different codons, of which 3 are stop codons that do not code for any amino acids, but only 20 amino acids. So one amino acid can be coded by more than one codon. Like in English, there is no limit to the sentence size, so many codons can be present in one sentence, open reading frame, ORF. The largest protein known is titin, which comprises about 27,000 amino acids and must have been coded by an ORF that is 27,000 codons long. If titin protein were to be read by the sequence of the amino acids, that would be, as some claim, the longest word in English. Bioinformation is physical in nature and essentially in a three-dimensional structure, which is determined by the sequence of nucleotides, in the case of nucleic acids, and amino acids, in the case of proteins. The 20 amino acids come in various sizes and electrochemical properties. Different side chains have different capacities to interact with other residues, water, and other ligands. These interactions lead to a stable conformation that may serve a function. The amino acid sequence gives the protein a structure from which a function arises that could eventually be useful for the propagation of the genome that encodes the protein. One protein can have multiple conformational states based on temperature, pH, and interacting partners, ligands. The ability of fluidity or dynamicity in conformational states with varying conditions makes life possible. All cellular life is based on the ability of proteins to change conformations. Different conformations confer different properties to proteins. These properties include enzymatic reactions, physical structures, and stabilities. These enzyme reactions and structures eventually make a cell that exhibits all the life properties. So, bioinformation is present in the form of sequences that may give rise to structural information, which aids in the manifestation of life. Sequence information precedes structural information. Let's get back to appreciating the fundamental nature of biological information. In all chemistry, no molecule dictates the formation of another identical molecule from building blocks except nucleic acids. Such a molecule is essential for the emergence of life. Material form precedes life, as seen in the organism's structure, such as the human body, and it emanates an identity. Identity exerted by the inherent properties of the material form lives on through time, which is the essence of life. To recapitulate the identity over and over requires the ability to replicate the information that can recreate the identity in the material form. It is funny to think of each of us, even all organisms, as the physical embodiment of information, or our genomes. We are walking info. That inherent ability of identity to exist in time despite the conditions is life. Unlike any other form of matter, nucleic acids are the only molecules that are matter, energy, and inheritable information. By virtue of specific hydrogen bonds between the complementary nitrogenous bases, information in one DNA molecule can induce the formation of a new molecule with similar information from its basic constituents. Guanine base pairs with cytosine through 3 hydrogen bonds. Adenine base pairs with thymine through 2 hydrogen bonds. Under normal conditions, these are the only stable base pairing combinations. Therefore, in a double-stranded DNA molecule, the nitrogenous base sequence dictates the nitrogenous base sequence in the complementary strand. That is referred to as the Watson-Crick base pairing. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1953. All bioinformation comes down to the structural basis of DNA and how information in one molecule can dictate the information in another molecule. This restrictive base pairing is essential for the recapitulation of bioinformation from one molecule to another. The sequence of the nitrogenous bases in one strand of nucleic acid is the information communicated through the base pairing through hydrogen bonding. Hydrogen bonds in the base pairing communicate, or determine, the complementary strand sequence, similar to the mold and idle pair we discussed earlier, but a double-stranded nucleic acid molecule can give rise to 2 identical double-stranded nucleic acid molecules. The sequence of nitrogenous bases is communicated, ensuring the formation of new double-stranded molecules. That replicability of matter gave rise to information that can be replicated. That is the genesis, literally, the let there be life moment. That is the beginning of biology. Everything related to the existence, interactions, and effects of entities with genetic information. Genesis started with intermolecular hydrogen bonding between the nitrogenous bases in nucleic acids. The hydrogen bonds are fragile, but these hydrogen bonds in the base pairing are something like the last missing cent one would require to buy a ticket to the origin of inheritable information, the origin of life, and a ticket for the Nobel Prize to Watson and Crick. If not for these base pairing hydrogen bonds, nucleic acids would not have been able to transfer information. They would have just been matter sans inheritable information. The emergence of base pairing is when information started. Everything from then on is what led to our ability to perceive information. Base pairing hydrogen bonding is the start of biological time because that is what led to our existence, and we are attempting to make sense of time and information. Life started with and is information. Life is the message, and the message is life. Biology entails every aspect since the first hydrogen bond that formed between the nitrogenous bases to humans and beyond.